So the translation, the initiation is the start of the polypeptide synthesis. And AUG is your major start codon. Sometimes GUG, but almost always AUG is your start codon with a methionine. That does not mean every protein starts with methionine. Later that can get chopped off. And in fact, in molecular biology, that's really important because the very first amino acid exposed can determine the lifespan of that protein. But during this process, in initiation, we're going to start with AUG. And once the correct starting point has been identified, a special initiator transfer RNA binds the ribosome. Elongation, you keep adding an amino acid one at a time, and it elongates in a linear fashion. An amino acyl tRNA enters the ribosome and delivers the amino acid. It attaches to the messenger RNA. The anti-codon on the tRNA will be complementary to the codon of the messenger RNA, and then you'll link in your amino acid. So the peptide bond forms between two adjacent amino acids, as shown here between glycine and methionine. The transfer RNA carrying the peptide chain moves over in the ribosome to the next amino acyl space. And the next amino acyl tRNA enters the ribosome, and the protein chain continues to grow as it comes in one by one. And then, once the transfer RNA has delivered its amino acid and it's empty, it leaves through the exit space. So at termination, polypeptide chain is terminated. So once you've added all your amino acids, let's say all 200 of them, you get one of the stop codons comes in. There were 3 stop codons. So a nonsense or a termination codon appears in the messenger RNA. UAA is one of them. If we go back to our code, you see we have 3, these TCs over here. These are termination codons. So that would be UAA, UAG, and UGA are our 3 termination codons. These are our stop codons, and they say stop. All right, so recall the sequence of the nucleotides in the messenger RNA, and this gives us our genetic code. So we can take 3 nucleotides, it's a codon, it stands for an amino acid. We can translate a messenger RNA into the proteins, into the amino acids that will link together to form the protein. So if you get UUG, we go back here. Here's the first nucleotide U, second U, G, that's a leucine. CAG, CAG is glutamine. ACU, we see ACU, threonine. And GGU, we have glycine. So we have leucine, glutamine, threonine, and glycine. So we can translate the codons into the amino acids. Mutations occur when there's a change. A new trait either appears in an individual that was not present in the parents or the ancestors. It is a new genetic or chromosomal change. Most changes are harmful. Every now and then you have a beneficial mutation which leads to evolution, but almost all mutations cause cancer or some disease and are harmful. Because mutations are genetic, they are passed on to future generations. And mutations occur spontaneously or are caused by chemical agents of various types like radiation, X-rays, cosmic rays, ultraviolet rays, or chemicals which are mutagens. Any agent that causes a mutation is a mutagen. So a mutation will change the sequence of nucleotides on a messenger RNA molecule. And notice our change is right here. This UGC becomes UCC. And this changes the product. What used to be a cysteine, UGC, is now a serine, UCC. So you've changed an amino acid on the protein, and that can totally change a protein. It can make it completely non-functional. It can be a devastating mutation. For example, sickle cell anemia, which should be a glutamic acid, is mutated into a valine. And just a change of one amino acid leads to all the symptoms of the disease of sickle cell anemia. And so one change can be a big thing in a protein. Genetic engineering. Here in a lab, we purposely change genes. And we can make species that have recombinant DNA, which means that in their chromosome you have the DNA of at least 2 different species. And we make a lot of pharmaceutical products this way, like insulin is made in E. coli. So it's a recombinant product because in the DNA for E. coli you have the gene to produce human insulin. It's from a different species. So important processes in genetic engineering, the DNA polymerase reaction when you make more DNA. The use of restriction endonucleases. These are like molecular scissors that can chop up your molecules. You can chop out a chunk and then you can add a chunk. DNA ligases are used to seal new chunks in. If you're genetically engineering a bacteria, you have to remember that a bacteria can only read coding information. It's not complex like a human cell. It can't read or remove introns. And so to successfully genetically engineer a bacteria, you first transcribe the human gene to a messenger RNA, and then you use reverse transcription, just like HIV uses, to make that messenger RNA back into DNA, because that's just the protein coding portion, and that's the segment that a bacteria can actually read. And then you use restriction endonucleases to chop open the DNA to open it up, and you'll insert your new DNA, and then DNA ligases will seal it up, and you have genetically modified your bacteria. And so that is for a biotechnology class. And that is the end of this chapter on DNA.